afternoon. Good afternoon. I'm on the screen, so I think it's started. Good afternoon. I'm Ruggero, and I'm just here to present you our speaker for today. First of all, I remind you that at the end of the speech there will be a Q&A session, and for those who are in Trieste, it's quite easy. I think it just you can ask questions by raising your hands. But for people who are following us online, they can find a specific function for the Q&A. Well, our speaker today is Professor Ada Yonath, and I'm going to read this just to get it right. She's the director of the Ellen and Milton Kimmelman Center for Biomolecular Structure and Assembly of the Weizmann Institute of Science, and in 2009 she was awarded the Nobel Prize for Chemistry, along with Professor Ramakrishnan and Stites, for studies on the structures and the function of ribosomes, which are macromolecular machines that can be found in our cells and that essentially transform what's written in our genetic code to proteins. And we did know what they were doing. We just didn't know how they were doing it. And so Professor Yonath developed a new approach, if you want, to crystallography, to understand how ribosomes are composed, what's their structure, and so how they work. It was a kind of research mainly driven by curiosity, and it was, if you want, knowledge for knowledge's sake, science, basic science at its finest. And at that time, it was seen as quite an impossible quest, a very hard quest to pursue, and a quest quite useless. We know what they do. What's the point in knowing anything more about ribosomes? And yet, I think someone can say that knowledge is usually inherently good, and that everything we can do to pursue more knowledge, more scientific culture must be pursued. But for the most pragmatic of you, we can also say that what started as a quest to just answer some of the most basic and fundamental questions about life now gives us a way to better understand how most of the antibiotics works. So, without any further ado, I'll present you our keynote speaker, Professor Ada Yonath. Good morning.
Morning. Thank you. Do you hear me?
Yes, yes, yes.
Okay. So, I'm happy to be here. Not physically, I would like to. Trieste is a beautiful city, and ESOF is a very important gathering. But at least from home I can, and I want to talk to you about how we went from basic science to modern medicine, and within it, about next generation novel eco-friendly antibiotics. And also, I have a blue dream, and maybe I can share it with you. So, I want to take advantage of this gathering to acknowledge the importance of the contribution of ERC, the European Community funding agency, to basic research. And I do it upon the transition from the funding program to the funding program Horizon Europe, which successes Horizon 2020. So I'm very thankful to ERC for our research, for their support in our research, basic research, and for supporting many others high-level, fantastic studies. So you already heard that ribosomes are important. They are translating the information that is in the DNA, the genetic code, via messenger RNA to proteins. And this is done in all cells. They perform the two functions, the decoding of the genetic code and the formation of the protein by making peptide bond. They do it in all cells the same way. They actually function in every living cell, in bacteria, in elephants, in cockroaches, in flowers. They are acting continuously, and they can form up to 40 peptide bonds. It means the bonds between the amino acids in 1 second. Mistake rate about 1 to half a million, or even some people say 1 to a million. Now, here I want to tell you something about myself. I was a very good student, the fastest in my class when I learned to make peptide bonds. Second year chemistry. And we had to make a single peptide bond, just one. And I was the fastest because I was very poor. I had many jobs and I wanted to finish with the lab tasks as fast as possible. You know, as fast as possible, the fastest took me 6 hours for one bond. And the ribosomes can make up to 40 bonds in 1 second. So how do they work? If we call them a factory, here is the factory, ribosome factory. It has 2 floors. The top floor reads the genetic code and decodes it. And in the lower floor, the amino acids are brought on carriers, which are also RNA molecules, tRNA. And when the code of this particular amino acid will be here, the triplet that codes for it, it will go in. And in the lower floor, this arrow actually happens in the lower floor. In the lower floor, this will be connected to the growing protein. The track will be empty. The message will progress by one codon, and the next amino acid will come in. That's all, very simple. So actually the 2 floors of it costs a little energy. It's very efficient, but some energy it costs. Actually, the ribosome, the 2 floors are describing the ribosome that has 2 subunits, small and large. All of them are made of 2 subunits, although they grew with evolution. The basic performance is the same. The small subunit, the top floor, decodes the genetic code, and the large subunit makes the bonds. So if this is the small subunit and the large subunit, the tRNA, this track that brought the amino acid, is doing the decoding here. And on the other side, it gives it to make the bond. That's all. That's so simple. But we didn't know how it happens. And now we do. And when we got the structures were just the tool to understand how the function goes, how the process continues. We made a little movie. Within a few weeks, we understood how the ribosome works. It took 20 years to get the structure, but then we understand. So here, what you can see here is the messenger RNA with the decoding, with the code information is reaching the small subunit. The 2 subunits are separating the cell unless they have to make a protein. And they learn that they have to make a protein by factors. And you will see in a minute one of these factors being connected to the small subunit here opens the gate for the entering messenger, and here in blue is the factor. This is factor 3, initiation factor 3. Now when the messenger is in place, the first track, the first RNA can come with another factor, initiation factor number 1, and put it in place. And when all of this is okay, the factors left and the large subunit can come and interact with the small one, makes bridges, and the active ribosome has been formed. And you can see it here from the solvent side. The top one is the large subunit. and it helps getting in and out the tRNAs. So they are brought by elongation factors and they come out empty. So we took away the large subunit. You can see how the decoding happens here on top in the small one. And from the large subunit, we left only the active site, or how activity happens, and the tunnel through which the newly born protein goes through. This is the protective way. So here is the tunnel in the large subunit. The protein comes out from the lower part, and the process continues until there is a stop codon. And then instead of elongation factors, there are factors that finish, that terminate the function. It's a stop codon, and it releases the two subunits. The protein, this blue thing, can go and do what it needs, and all the other participants are waiting for the next protein to be made. Very simple, very elegant. So just to show you that in the movie you couldn't see details, but actually we know the position and the coordinate of every atom. In the beginning, and only in bacteria, now also in higher organisms. Ribosomes are made of the two subunits. They are mainly of RNA, made of RNA, and many, many proteins. Each protein is shown here in another color, the RNA in gray. So just to remind you, the small subunit is doing the decoding, and the large is doing peptide bond, makes the peptide bond, forms the bond. So the small subunit is also called SSU, small subunit, and the large subunit. And that's correct for everything, all ribosomes. Also, I would like to tell you that there is a huge number of ribosomes that function in each living cell. Mammalian cell may contain up to millions of ribosomes, like in the liver, for instance. Even bacteria cell can have, some bacteria can have up to 100,000 ribosomes in a single cell of bacteria. So owing to the vital role played by the ribosomes, over 40% of the clinically useful antibiotics target protein biosynthesis. So if you want to kill a bad bacteria, it's very clever to stop the production of proteins. So this is what is done by over 40% of the clinically useful antibiotics today. They are targeting protein biosynthesis, mostly by paralyzing the ribosome. So just to let you, whoever doesn't know, the natural antibiotics are natural, made by bacteria. The weapons that one microorganism, let's say bacteria, from one type are using for interfering with cell life of different species. It's the war between bacteria. And the antibiotics are doing it, as I said earlier, 40% of them, they are doing it by binding to the active sites. And you will see here four examples. This is adenine, and it just blocks the entrance of the messenger RNA with the code. That's all. So small, so clever. That's no code, no nothing to do. The second one is even smaller, and it's called tetracycline. It's more useful medically, and it blocks, or it associates with the position of the tRNA, the second tRNA with the amino acid. So one tRNA is not enough because there will not be a bond. That's it. And the next one that we'll see is the first one to get used for ribosome antibiotics right after penicillin. It's called erythromycin, and it blocks the tunnel for which the protein, newly born protein, has to go. So it can be made, few bonds can be made, but most of the proteins won't go on. And the last one is the cleverest chemically. It just goes into the peptide bond, just sits there. So bacteria against bacteria, They are very clever. And just to show you again, the same what you saw in the movie, just to say it here. You see the two subunits, the large on the left and the small on the right, and many balls, and each ball represents a family of antibiotics that they are sitting in the decoding, in the bonding, in the hinging, and in the tunneling, and many other positions, in the entrance of the amino acids and so on. Pharma companies, when antibiotics came in, got really very excited, and they made the improvement. They improved the properties of the natural antibiotics. So here is a macrolide, which is the family that came out from erythromycin. You can see on the left the backbone, and you can see all types of additional things here. So they are actually extensions of the erythromycin scaffold. This is what the companies did very actively when erythromycin was found in the middle of last century for about 20, maybe 30 years. But the question that we should ask is, since the ribosomes active sites are highly conserved, as I said, they do it almost the same way, actually the same way in all evolutionary entities, how the selectivity is used, because we want to kill the bad bacteria, the pathogens, not the patient. So the clinical usefulness is based on very small changes, very small differences. So you can see here a cut, vertical cut through the large subunit with one tRNA at the active site and the tunnel through which the protein will go, and the position really near the position where proteins are being made, the position that is blocked by erythromycin. So we looked at the tunnel this way, or if you want to draw it just for direction, this is the tunnel, and we looked at here. So what you see here is a cut through the walls of the tunnel. They are all made of RNA, gray, and inside here you can see several antibiotics from the erythromycin family, from the macrolide family. If you want, this is the chemical formula of erythromycin. And they were extended, as I showed earlier, or substituted by companies. They all sit in the tunnel. They all block the tunnel. Some are blocking excellently. Some are blocking very good. Some are just blocking, but they are blocking, or almost fully blocking. And all interact with one of the components of the tunnel wall, which is adenine in position 2058. This is the way it is in bacteria. So here is erythromycin, and here is this adenine. Just that you can see that the contact here, the affinity here is very high, as if a very, very good bioinformatic or chemical person, chemist made it. But it's not, it's by bacteria. And this is it, how erythromycin binds to adenine in position 2058. In human, in mammalian, in eukaryotes, in all eukaryotes, there is no adenine in this position. There is a guanine. That's all. The difference between adenine and guanine, this is the difference. But when there is this piece, is here, the guanine piece, the distance to the erythromycin is too short, and there is repulsion, so it doesn't bind. That's all between us and pathogens or bacteria. So I showed you how the selectivity works, but we also learned how resistance is happening. And just before this, I want to say, and I'm sure that you all agree with me, that it's one of the most severe problems in modern medicine. It was shown that resistance is also bacteria against bacteria, or bacteria saving itself by walking to the jungles of Venezuela, to places that there was not any European or American medicine, and they still found genes for resistance. So it's bacterial survival. The bacteria profit from accidental mutations. Sometimes they even develop specific molecular pathways that cause resistance. I won't talk about all this now. I'll just show that one of the processes, one of the ways that it's being made, you remember selectivity was based on A at position 2058 versus G that did not allow binding. So for bacteria that can mutate, maybe they mutate zillions of times, but the resistance is acquired by this mutation from A to G, that's it. And this is one of the pathways of resistance. It's not the only one, it's even not the most popular one, but it's very clear how it works, and it's a good example. So the increasing appearance of multi-drug resistance, many bacteria are resistant to many drugs, together with the minimal, actually negligible number of new antibiotic drugs that are presently undergoing development, is becoming a colossal, a global health threat. I'll give you some examples. In USA, between 2010 to 2014, millions of people fall ill in infectious diseases. Most of them did not recover every year. In Europe, in the same time, between 2010 and 2014, resistance caused 3,300 deaths. So sorry, 33,000 deaths every year. And it also cost, until the end of it, 1.6 billion euros for healthcare. And it also required productivity loss, although the consumptions of every specific antibiotic has almost doubled, yet it was not useful for these 33,000 people dying yearly. So it seems that we will soon revert back to the pre-antibiotic era, during which diseases caused by parasites or by simple or severe infections like pneumonia or wounds were almost untreatable and resulted in frequent deaths. And the World Health Organization announced that we are now in the post-antibiotic era, and consequently, the World Bank estimated that up to 3.8% of the global economy will be lost by 2050 just because of resistance. It's a huge, huge, huge loss. Yet, only a few antibiotics are in development. I said it earlier, and now I'll show you something about it. Most drug companies, most large and famous drug companies stopped attempting at creating new antibiotics. Some smaller ones are trying, but very little, you will see in a minute. Owing to the huge mismatch, because the social value, it means our life, our health, and the high expense needed for creating new antibiotics. So you can see here on the left side, the increase of drug resistance until 2002. So three different bacteria and their resistance. And here on the right, you can see the number of new antibiotics until more or less 2007 and 8, something like this. 1 year 4, 1 year 2, that's it. It goes down, and as I found last year, there was now none new. So the question is, if companies don't do it, and we need it, the question is, can we really combat resistance antibiotics in full? Well, in my opinion, it's unlikely, because bacteria want to live, want in parentheses, and because bacteria are cleverer than us, at least in terms of survival. Yet, we decided to do some of this contribution, maybe not giving full fight for resistance, but combating it and getting better and less resistance and more clever resistance, more clever fight against resistance. So what were our beginning point or our assumptions? Since only resistance bacteria continue to proliferate in the presence of antibiotics and increase in number over time, hence using wide spectrum antibiotics, the end result is a population of more resistance. Because for a disease, we need a specific one, not all the wide spectrum. And those that are not used are causing resistance, that are not needed, sorry, for the particular infection, they are also causing resistance. So first assumption was using narrow spectrum antibiotics. This will narrow the risk of resistance, reduce the risk of resistance. Second, our concept for fighting or controlling bacteria's wisdom is identified pathogen-specific, as I said a minute ago, essential structural motifs on the ribosome's periphery. Why periphery? This is far from the active site. Therefore, bacteria did not learn yet to approach these positions. So for this reason, we determined the structure of ribosomes from pathogenic bacteria. We have 4 or 5, but now the 5th came, and compared them to ribosomes from harmless bacteria. And we had lots of lessons. General lesson, all bacterial ribosomes are similar. But now specific, lesson 1, lesson 2, lesson 3, I save you. I show you lesson 4, because this was beyond expectations. And here, what is beyond expectation? Novel antibiotic binding sites. Those that were not known before and are not known yet to bacteria itself. So if you look here, you can see in gray the structure of the large subunit from a non-pathogenic bacteria. And this blue, the cyan pieces, are additions that are on the pathogen, here, 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 and here. This, in our opinion, can be sites. So let's focus on one of them, this one. And here it is. You can see that most of the structures are the same, but here there is a little addition which is pathogen-specific. So we think it can be a potential binding site. Or if we look at it together, this is the structure. It's very similar except for these positions. The one that we just looked at here, which is a potential binding site. I hope you agree with me. The bacteria is Staph aureus. And actually there are more positions. All these can be. So in fact, in the model system, we identified 25 new potential unique sites. We block them one by one, and 16 of them inhibit protein biosynthesis. So these sites are not involved in the primary ribosomal activity, not in the decoding, not in peptide bond formation. Hence, so far, no pathogen contains genes for their modification. This means that so far, no bacteria assign vital roles for these sites. Therefore, for now, these sites can be exploited for the design of advanced degradable antibiotics, hence environmental friendly. So I will talk about it in a minute. I just want to say that these sites are important for the ribosome interaction between themselves or with the factors and so on, but not in the active site. These are the sites that we found and can be targeted for blocking activity. So what do I mean by degradable antibiotics and environmental friendly? So first, before I go on, I just want to show you that we have several sites, 25, but I show here 5 of them that has very nice, very impressive IC50. It means power of stopping the ribosome. So let's come now to selectivity and similarities. First, about the microbiome. I'm sure you know that there is microbiome in the flora of our body and are very important. They usually are, most of them are in the guts, in the ear, in the lung, in the skin, and so on. And most of them are extremely useful. They are mainly eubacteria. It means they have mainly almost similar, maybe even similar active sites. So essentially, the antibiotics that we are using for the pathogens can reach them. If they can reach them, they can even get rid of them or harm them. And this is actually what happens. In many times, one comes with a small problem, infection, so after a operation, some dirt around the places that have to heal, and they come out with a real disease that is caused by knocking out part of the microbiome. Second thing is, are these sites okay for using in mammalians? So here are the ribosome surfaces in mammalian compared to bacteria. Bacteria in gray, and in mammalian, all these additions. I don't want to go to explain which one is what, but you see that most of the differences are on the surface. It means that the new antibiotics are fully safe for the mammalian ribosomes because the surface is very different and we are working on the surface. So let's go to the next lesson about environmental and ecological consideration. So most of the ribosomes, ribosomal antibiotics, are extensions of small organic compounds. You can see here the organic compounds. There are usually some chemistry around them. And these compounds, this course of the antibiotics cannot be digested by eukaryotes. So, and they cannot be degradable in the environment. It means they are not eco-friendly. They are contaminating the environment. But ours, sorry, I want to go one more. They can go even back to us because some of them are not detected by the purification system since they are so small, and they can go into irrigation system and come back to us. But the inhibition of protein biosynthesis by the newly identified potential site by us. You remember I showed you these red things that can be on the sites. So these are molecules that we are determining, we are designing. So they are composed of nucleic acids or oligo-peptides, or the two of them. They are fully degradable, and they can be optimized in their chemical properties, in their antibiotics action and degradability. So because it's our chemistry, we can call them, even for poisoning. So before I go on, I just want to repeat that over 40% of the clinical useful antibiotics target biosynthesis, but because of the vital role played by ribosomes, there is another way that cells develop methods for preservation themselves, like under a stress condition, the cells want to keep their ribosomes. They don't do many new proteins. Maybe the stress is hunger for amino acid or something like this, any stress. But meanwhile, they don't want to get rid of their ribosomes. Ribosomes will fall apart if they don't protect them. So the bacteria and also the higher organisms developed a way to combine between pairs of ribosomes. You can see here a ribosome, large subunit, small and large, that sits in a way that one is blocking the entrance of amino acids or messenger RNA, and they are this way, they are preserving their ribosomes. So when the problem is over, they have active ribosomes. They open here the contact, and there is a functional ribosome. This way of dimerization is different in different bacteria. Here we call it hibrination, and here there are 3 different bacteria, and we put one top of them. You can see that they don't look the same. So we are designing antibiotics to the specific positions of the pathogens among these all bacteria. So in summary, the new insights obtained from these high resolution structures of the ribosome from a genuine pathogen provide unique chemical tools for suggesting novel sites for potential future antibiotics. I showed it. For suggesting how to improve clinical performance, I didn't show, but we found a way to do this too. And for better distinction between pathogens and useful bacteria, bacterial species in the microbiome. So the pathogen-specific antibiotics means design of antibiotics, drugs specific for each and every pathogen, in contrast to the current preference of broad-range antibiotics. So we are aware of the problematic economical issues associated with pathogen-specific antibiotics. There are problems even with the easier one, with the wide range. Nevertheless, we expect pharma companies to consider human aspects, not only profit. Human aspects are also for the companies. Okay, in short, our structure-based specific antibiotics design aim at minimizing widespread resistance, microbiome destruction, and environmental hazards. We don't think we can get rid of these bad things, but minimizing we can, and this is why we work. Moreover, in the currently, we are extending our studies towards exploiting structure-specific motifs, technology for treating human diseases that are connected to ribosome maptis diseases, which are associated with minor modifications in the ribosomes. And these are mainly cancers and anemia, but not only. So the same idea is for the mutated ribosomes. We look for specific motifs, and the technology is to target them. So I promised you a blue dream. My blue dream is connected to life. So this is the way life expectation was in the whole world about 15 years ago. You can see that the life expectation over 80 or over 75 was in America, in Europe, including Italy, in Israel, in Australia. Then not so great, but still acceptable in Chile, in part of Brazil, in part of the Middle East, and even 65 years here in Russia, between 65 to 70 years. But there are still places in the world that are less than 50, less than 60, all Africa, and positions here. So my blue dream is this, the whole world in the blue. And for materializing my blue dream, a serious commitment of pharma companies is essential now. So I talked about basic science, and I finished with antibiotics. I want to come back to basic science. So structural studies in order to understand function started first in England and then in America in about the 30s, actually the 40s. And it led to many, many very important structures and obviously functions, and many Nobel Prizes. All these are Nobel Prizes. Where am I? Can you find me here? Don't look fast. I'm here on the floor. Treeless. There are only 4 treeless and I'm part of it. I grew it, I increased it. You can see here on the floor. And why do I show it to you? Because the question is, how did the treeless basic scientist reach the above insights, these insights I told you? By support, first of NIH, and then when ERC started, of ERC and ERC. And I started with thanking the ERC for supporting basic science, and I want to finish this way. I also want to thank the Weizmann Institute that let me work, although for over a decade, 15 years, I was considered a dreamer and a fantasiumer. The decision was made by the president, Michael Seller, and he was supported by Kendra and Adfitzen, 2 Nobel Prize winners for the structure of myoglobin and for folding of proteins. And by Alex Rich, they tried to do the same way work we did to determine the structure of ribosomes for over 10 years and failed, but still was supported. It all started in Berlin with a very strong collaboration with Professor Weizmann, the Max Planck for Molecular Genetics, who was also very important in starting a group for us near the synchrotron where we collected the data. He died 10 years before the structures came and was replaced by Franceschi and then by Faccini. So I had two groups, one in Hamburg and one in Israel, and I want to thank all of them. This is the group in Hamburg that was terminated in 2040. The technicians say that only angels can work on ribosomes. Here they came to Israel, to the Dead Sea to look for good bacteria. And here they had a birthday and they made some birds, so they had to clean. And this is the group in Israel that I'm showing the people that have been there through the last 10 years. And those that are still in are with this green star. So the group looks large, but actually at every minute there are few. Here are the places we collect the data, all the synchrotrons, and who funds us, again, NIH, ERC, and also Max Planck. I want to show you Tamar. Tamar came to us for 10 weeks, 20 years ago. She's still around. And she had a birthday the day I took the picture. So she baked a cake. Can you see her cake? Maybe not, so I grew it up. I increased it. And this just shows that for our group, the ribosomes are considered sweet. I also want to thank my family that we are supportive. Here you can see my daughter and granddaughter, but also my mother, my sister, and so on supported me, although I was disappearing a lot for experiments and so on. And I want to tell you what my granddaughter said when I got a prize in Paris. She said, as you know about me, she is a very busy scientist, but she always finds time for me. And she invited me at the age of 5 to the kindergarten, but that's not important. What's important, she gave me a prize. That's not her prize, this is the Nobel. This is her prize. That for me, it's as important, maybe even more important than the Nobel. The grandma of the year is Ada Yonat. She was 9 years old at that time, and I asked her, which year? The grandma of the year, which year? Every prize is a year. No, she said, you have to reprove yourself every year. And if you fail, I take it off the wall, still on the wall. You see? So ribosomes became popular in Israel. This is the carnival. The kids became ribosomes. And he became me, and his wife is here, a ribosome. You see, they are dancing. She is the production of this protein that comes out. And this is how our one of our best, most beloved writer and artist made a head full of ribosomes. And if you look closely, small subunit, large subunit, large subunit, small subunit, and completely asymmetric. And you see here, and here, and here, and here. This is where he thinks we should put new antibiotics. Thank you.
Thank you so much. Thank you so much. I've just been told that we have got some more minutes for the Q&A, and that the first thing I said was wrong. So if people here in Trieste want to ask questions, they do not just have to raise their hands, but there are 2 microphones that they can use. We have a question coming from people who are following us online, and I'll try to read it if you can hear me. Yes.
I can hear you, yes.
Okay, perfect. It says that new research shows that ribosomal environment is key for their function, and that regulatory proteins seems to stop or start the ribosomal activity. He just wanted to know if you have any insights on this kind of research.
Of starting and stopping and so on.
Yes.
There are now many studies in many labs. Even last week, a new one from Ramakrishnan's lab of starting and stopping in bacteria and in higher animals, even in human. If you write me an email, I'll send you references.
Perfect. Thank you very much. And then I was wondering, just to break the ice, since there is a question. Okay, there's you can go to the microphone and ask it. Perfect.
Hello, can you hear me?
Yes.
So thank you so much for this very inspiring talk, including the very last part, which is touching, actually. So I'm not a biologist. I was kind of impressed and scared by your comment that bacteria are much cleverer than we are. And seems to me that COVID tells more or less the same story. It's just a little piece of crystal which can kill us. So the question is perhaps a bit philosophical, but nevertheless, is it that at some point our complexity is a sort of liability towards these pathogens whose strength is just their simplicity? How would you comment on that?
It's a very interesting question, and sometimes I think about it. And let me actually go back to life in order to partially relate to this question, to your question. It's not an answer, it's just relating. The virus that we have now, COVID-19, doesn't have a ribosome. It cannot reproduce itself. It needs a host for it.
Yes.
In this particular case, a mammalian host.
Yes.
They say that it came from another animal, but anyway, whatever it is, it needs a host. It's so clever not only to enter the cells of the host. For this, it has a spike and a cut. This is more mechanical, if you want, partially mechanical. But then it has the power to conquer the ribosome of the host, not only one, many of them, in order to replicate itself. Okay. So it was already shown that it has in his genetic code, it has a code for making a protein that stops the ribosome, that can go into the path of the messenger and block it.
I see.
Okay, so we know how it stops, but we want to know how it convinced the ribosome to do his protein. And this I don't understand.
Well, you will
very,
soon.
Very
Okay, I hope you will soon. Thank you so much. Thank you again.
We got another question?
Hello, Dr. Yonah. Thank you very much for your fascinating presentation. I'm not really a biologist either, and I have to say that one of the more alarming things I saw in your presentation is how under-researched novel antibiotics are, and I was wondering if you could please expand a little bit on why that is.
I'm sorry, I didn't understand what you're asking.
Why isn't there more research on novel antibiotics?
Or because mainly companies, but also scientists, we all know that there will be resistance, even in the sites that we suggest that so far the bacteria don't understand that they are important. With time it will come. We think that because we have so many new sites, and if the doctors will agree to use one at a time, and it takes about 10 years, 15 years for strong resistance, that maybe if we have 10 of them, we have 150 years and long enough. But we didn't show it yet, we are working on it, and the companies cannot take this risk because they have to show profit. I hope, and I imagine that they will change their mind when they will see that there are not enough old people to buy their expensive drugs because they die earlier from infections. But I'm not sure. But companies don't take this risk because at the moment they have to show profit. And scientists, young scientists, or just scientists, there is some research, except for ours, maybe 5 to 15 labs in the world. I think not enough because the chances to win are not very high, although they are very needed. So I, all my life, worked on what I wanted to understand, what is needed for society, and didn't really look at chances. But some other people are more sensitive to how the world is looking at them.
Thank you. Thank you. Thank you very much. Our time, unfortunately, has ended, and I would like to thank you again, Professor. And it was a really interesting keynote to hear. And thank you very much.